Skeletal remains have been historically overlooked in archaeological contexts. The additional disregard of commingled, fragmentary, and non-adult bones have led to entire areas of prehistory going unnoticed. Modern bioarchaeological research aims to rectify that. Even though commingled and fragmentary remains provide many challenges, conducting research on them gives us the ability to gain further insight into the past, and in doing so, the present. My name is Christian Berends. I'm a junior studying at the University of Michigan, majoring in anthropology with a focus on the Middle East.
And I'm Rachael Orkin, a sophomore at Los Angeles Pierce College majoring in microbiology. We have had the privilege of taking part in a summer research program funded by the National Science Foundation called Research Experiences for Undergraduates, or REU. As part of our research, we examined commingled and fragmentary skeletons from the an-Nar period of the United Arab Emirates in an effort to examine past demographic patterns.
The an-Nar period dates between 2700 and 2000 BCE and includes above-ground circular tombs found throughout southeastern Arabia. These tombs were used for hundreds of years during the early Bronze Age, with some tombs containing hundreds of individuals. We have examined skeletal remains from tombs an-Nar 1 and an-Nar 2 from the Emirate of Ras Al Khaimah.
By using current bioarchaeological methods for measuring unfused fetal and non-adult basilar portions from the occipital bone of the skull, we sought to estimate age at death. By doing so, we were able to test hypotheses that can help further our understanding of these ancient communities, and more specifically, whether fetal or other non-adult age groups were allowed to be interred within these Bronze Age tombs. When looking at other an-Nar sites around Arabia, such as Tel Abrak and Al Sufa, we found that fetuses were allowed to be interred with adults. Therefore, we hypothesized that at an-Nar 1 and an-Nar 2, this would also be true.
We also hypothesized that an-Nar 1 would hold a greater number of fetuses and infant remains than an-Nar 2. While the use of both tombs overlapped during the late 3rd millennium BCE, an-Nar 2 may have been used first, and thus may have had dates that more closely coincide with the initial shift to a more sedentary lifestyle as a result of the adoption of oasis agriculture. This new subsistence strategy provided shade for crops from the leaves of domesticated date palms. Early on, the inhabitants of the region would have subsisted on a more varied diet consisting not only of these new crops, but also would have continued to rely on herding goats and cattle, gathering marine resources, and hunting wild animals. Only later would these communities become more reliant on agriculture and thus have less varied diets. We hypothesized that because this earlier period had a greater varied diet, this would have promoted better health than in later years as agriculture and sedentism intensified. Therefore, an-Nar 1 would have a higher number of fetuses than an-Nar 2.
The basilar portion of the occipital bone fuses to the lateral portions of the occipital bone at around 4 years of age, and as the basilar grows over time, its changing size can be reliably associated with certain age groups. We used methods outlined by 3 different groups of scholars to estimate age by measuring different dimensions of this bone.
Our primary 2 measurements included the sagittal length and the maximum width of the basilar portion, which we took using digital calipers. We then compared our measurements to published data tables and plugged them into statistical formulae to establish the ages of these individuals. Additionally, a 2017 forensic study by authors Olivares and Aguilera suggested that age could also be estimated by taking measurements of areas on the basilar that eventually fuse with the sphenoid bone and other parts of the occipital bone. This was very helpful due to the number of damaged basilar portions in our assemblage that were missing areas and needed to measure sagittal length or maximum width.
Through our analysis of basilar measurements, we found that regardless of time period, fetal remains were interred in both tombs. By the inclusion in these tombs throughout the 3rd millennium, this shows us that fetuses were already considered to be valued members of their surrounding community and thus eligible for interment alongside older non-adults and adults. This means that despite changing subsistence strategies that influence the social organization of these communities over time, fetuses were consistently permitted access to these tombs. Due to small sample sizes, however, we were unable to test whether there was a statistical difference in the number of fetal individuals between an-Nar 1 and an-Nar 2.
We also found that the different methods used in this analysis produced variable age estimates. Some were similar to one another, while others produced different age ranges. Because of this, depending on the method and measurements used, Our results suggest that between 20 and 50% of non-adults below the age of 5 were fetuses. The frequency of fetal remains at the site were similar to those reported at nearby an-Nar site of Tel Brak, where 34% of non-adult remains under the age of 6 were fetal, but higher than Al-Sufa, where 8% of individuals below the age of 9 were fetal.
Over the course of the summer, we were able to learn many things about the an-Nar period of southeastern Arabia through the context of tomb membership. We found that fetuses were deemed to be members of the community, highlighting a shared worldview within the region, as other an-Nar sites also included fetal, perinatal, and other young adults in their mortuary structures.
We would like to thank Dr. Ullinger and Dr. Gregorica for working with us this summer, without whom this project would not be possible. We would also like to thank our guest project mentors, Dr. Sharon DeWitt and Dr. Dwayne Bocot, who lent us their time to support our learning, the National Science Foundation for funding this experience, and the government of Ras Al Khaimah for giving us the opportunity to research the an-Nar 1 and 2 human skeletal remains.
Thank you for an incredible experience, and we will be looking forward to future findings from this region of the world.